Good day, everyone. Happy New Year and welcome. I'm Jack Van Horn from the University of Virginia, and please allow me to welcome you to this edition of our 2020-2021 Foundations of Biomedical Data Science seminar series. This lecture is supported by UVA's Integrated Translational Health Institute of Virginia, the UVA Brain Institute, the College of Arts and Sciences, the School of Data Science, and through a grant from the National Institutes of General Medical Sciences. It is my delight and privilege to welcome our speaker today, Dr. Kevin Pelphrey, Professor of Neurology from here at the University of Virginia. Dr. Pelphrey is a neuroscientist and expert on brain development, and he is the Harrison Wood Jefferson Scholars Foundation Professor of Neurology, and also the Director of the Center for Healthy Brain Development here at UVA. He earned his PhD at the University of North Carolina in Chapel Hill in 2001. And since he's had extensive experience teaching and conducting research at leading institutions such as Carnegie Mellon, at Duke University, as well as at Yale. Before coming to UVA, Dr. Pelphrey served as the Carbonell Family Professor in Autism and Neurodevelopmental Disorders at George Washington University, as well as the founding director of the Autism and Neurodevelopmental Disorders Institute. His team seeks to better understand the causes of autism spectrum disorder and related neurodevelopmental disorders to deliver the right treatment to the right individuals at the right time. Kevin is a principal investigator of the NIH Autism Center of Excellence program, in particular, the Neurogenetics of Autism Network. He's also served by appointment of the Secretary of Health and Human Services as a member of the Interagency Autism Coordinating Committee. His presentation today is entitled Building Precision Medicine for Autisms, plural. As always, we are streaming this lecture live and for recording via YouTube. And if you are watching via YouTube, thank you so much for joining us today. Also, our specially selected 2020-2021 Biomedical Data Science Innovation Lab participants are encouraged to submit any questions for Dr. Pelphrey via the chat feature in their Zoom sessions. I will personally synthesize these questions, collect them, and ask them on your behalf during the last 10 minutes or so of the lecture. And with that, welcome, Kevin. Thank you so much. We are very excited to hear your lecture.
Thank you. Thank you very much. And thank you for those who are watching. I'm very excited to do this. Excited to view a lot of the other presentations that are being done. I think it's a great resource. So I'm going to talk about autism today. And autism is a condition that currently is behaviorally defined. Think of making a behavioral diagnosis of autism when we see that a child or a young person, young adult, has qualitative differences in social functioning combined with restricted and repetitive behaviors and a rigidity of thought that's characteristic of autism. And when we see those things together and a developmental history of those differences in functioning, then we make the call autism. However, there's vast heterogeneity in terms of functioning and adaptive behaviors and severity of symptoms across a wide spectrum of autistic people. And that makes it very difficult to really plan for the proper interventions and the right types of treatments that we want to select. And so, fundamentally, the fact that we still rely on observation and behavioral diagnosis and developmental history, as opposed to what has happened in other areas of medicine, where we have more biologically informed diagnoses that refer back to the cause or the causes of a condition. So we think about the cause, and we know there are multiple causes of autism, and depending on the biological mechanisms involved, you get this difference in presentation that we think of as the heterogeneity on the autism spectrum. So this kind of lack of biological markers and our inability to more precisely diagnose autisms, now plural, thinking about the different causes, leads to a status quo where we have inexact treatments. We tend to apply the same evidence-based interventions across the spectrum, and they work better or worse for different kids for reasons that we don't really understand. So we have this broad application of inexact treatments, and that can lead to a lot of wasted time, a lot of failures to optimize progress, a lot of loss of resources for families who are paying for these interventions, and in general, most importantly, a lack of helping the child and helping the child's family to optimize the outcomes for the kids. So a lot of the work that I want to show you today is aimed at trying to understand the different causes of autism and trying to pursue different biological markers that might help us directly address that problem of a lack of mechanistically informed indicators and diagnostic instruments for autism, as well as prognostic indicators of what would work best for which kids, all right? So something that I want to emphasize from the outset is that while our work is translational in nature, and we're hoping that the work that we do will have a direct impact on families, it's built upon almost a decade now of really basic science that has advanced our understanding of different components of the human brain and how those components interact. So in the first editions of the kind of Bible of neuroscience, principles of neuroscience, there were chapters devoted to cognition, to language, to motor function, but little mention of social functioning, which seems kind of funny now when we think about it, because that's such an important component of being human. How we interact with other people, how we understand other people's intentions, and how we understand other people's perspectives. But around the late 1990s, early 2000s, there was really a revolution in neuroscience, cognitive neuroscience, using different imaging techniques to start to understand that there was a chapter yet to be written on the social brain. And so the concept of the social brain is that we have dedicated systems for processing the actions and intentions of other people, and that we can very rapidly and readily use these dedicated systems. They're modules, but they're interconnected with other systems that do other things: language, higher order reasoning, executive function, even our action systems for motor responses. And they're central to our understanding of other people, and then informing the rest of our brain as to what actions or what words we want to use in order to respond to kind of a complex to and fro with other individuals. And one of the little bits of the social brain that will feature prominently today, just one component that plays an important role in what now is understood as a widely distributed system, is the superior temporal sulcus region. And this is a part of the brain that processes what we call biological or social motion. And this is a little movie you're seeing now. Back in the early 2000s was high-end virtual reality. Now it's a little bit of a joke when we talk about virtual reality compared to what we can do now. But it was a simple experiment where we were showing in typically developing college students different types of motion, be they people moving or even a robot moving like a person, compared to, for example, a little caricature of a grandfather clock or a mixed up robot that was upside down and the parts were all mixed up so that you didn't see it as a person walking. And what this early study showed was that there were regions of the brain, especially along the temporal lobe, just above your right, mostly in the right hemisphere, just above your ear, that respond quite strongly to biological motion and are dedicated to processing that type of motion, but don't respond to other types of motion. And so that was an early demonstration that the brain, already just in seeing people moving, starts to treat other people in the environment differently than it does other moving objects in the environment and other types of visual information coming in. So it tags it as having special significance and applies a whole set of computations to it that aren't used for other stimuli. And what's really striking about this is that even from the first days of postnatal life, human infants and actually a lot of other species treat the movements of their species. So humans look at human movement already from birth very differently than they do other types of movement. It's as though they're pre-programmed by a combination of genetics and prenatal experience to hone in on that type of motion and see it as unique and interesting and learn from it, something that allows them to progress through their social development. So I'm excited now that there's actually a chapter on the social brain in the new Principles of Neuroscience textbook. So we've come a long way, and it's that basic science that really supports our efforts to look for biological markers. So one of the earliest studies we did of children with autism, we're putting them inside the MRI magnet. We were taking pictures of their brain as they were watching either coherent biological motion, this is a good example of that. So they're watching these little point light displays, they're called. These have been used in psychology for many decades to study, how we process motion. And then, more recently, once MRI came along and starting to be used a lot in cognitive neuroscience, started using these stimuli to study social perception. See, in this case, you have a person being filmed doing patty cake, actually, so sort of a child-friendly interaction. And then the scrambled biological or non-social motion, now I'm playing that next to it, and that's just the same exact point lights, but now scrambled up in a way that hopefully no one would recognize as socially meaningful, even though you might recognize it as somewhat equally interesting because it's moving around in a kind of random way. And so we had these two stimuli, and we would show versions of these different actions and different scrambled actions to kids with autism while they were in the magnet. And we did this study on a large number of participants over time. And so at some point, we started using the brain scans that would result from this in a much more data science oriented way. And we did some machine learning to see if we could use the patterns of activity that we would see from kids watching these different types of movies across several different studies. So typically developing kids and kids with autism, to see if we could go in and start to identify, what are the parts of the brain and how they're interacting that best separate individuals with autism from typically developing individuals in a diagnostic sense. And so we did this using some machine learning algorithms where we first had a discovery set. So we had, known the computer, we were telling it, okay, these are kids without autism, and these are kids with autism. Hopefully you're able to see my mouse there moving around. And we're saying, okay, learn the different features of the data set, these imaging voxels, and their activity over time, so in essentially four-dimensional space, including time, where we could differentiate the kids with and without autism. And the computer algorithm is learning up, how to do this. And then we used a much larger replication data set by virtue of doing this type of scanning over a long period of time with many kids. And we said, okay, now we're not going to give you any hints about who has the diagnosis and who doesn't. And we want the computer to apply what it learned and see how well it does in a new data set. And the results, I think, were really intriguing. So it leads you through this. So panel A here, panel A is showing you the parts of the brain that best differentiate kids with and without autism, okay? And these are components that we have come to be called the fusiform face area, fusiform gyrus, lateral fusiform gyrus to be specific, and the part of the brain I already mentioned, this right superior temporal sulcus region. These were the regions that, statistically speaking, did the best job at differentiating kids with and without autism. Now I want to bring you down to C here for a moment. You notice this says all boys, and this is a receiving operator characteristic curve. And these are often used to display data that we want to use for making diagnostic decisions in medicine. And what this is suggesting is that our model did quite a good job, area under the curve here of 0.77, of differentiating boys with autism from typically developing boys. And if you're either an expert on this type of data, or you're carefully listening to my words, you notice I went from kids with autism to boys with autism. So when you look up here at panel B, you'll notice that for girls with and without autism, our model did a terrible job at differentiating girls with autism from typically developing girls. In fact, it almost, it did do worse than chance, which is kind of hard to do. It's either we broke math, or the social brain is actually more active and more connected in girls with autism than typically developing girls, which is something we'll return to in a few minutes. And so on the one hand, we were very excited that we had an example of a potential biological marker that could distinguish kids with and without autism. But on the other hand, we were disappointed that it seemed to only work for boys. And then at the same time, we're very intrigued as to why it doesn't work for girls. And this was in the greater context of findings within genetics that girls with autism, in order to receive the diagnosis of autism, needed to have a stronger or larger genetic hit in order to get to the same behavioral diagnosis. At the same time, in that context, was knowledge that oftentimes girls were going undiagnosed until later in development. Oftentimes, whereas the average for diagnosing a boy with autism, let's say, might be 2 to 3 years of age, for a girl was pushed back at least a couple of years to 4 or 5. And there were many clinical anecdotes of girls who were really not getting diagnosed until the early, late preteen, early teen years, where they were really facing greater social challenges at school that kind of led to them getting in some disciplinary trouble. And then upon kind of really being noticed at that point, we're getting the type of diagnostic care that led to a diagnosis of autism. And it wasn't that they had become autistic later, they were always autistic. It's just that our diagnostic process had really missed them, and they hadn't been brought to the attention of people who could do a reliable diagnosis. And so they were the girls with autism in general are at much greater risk for suffering from the effects of getting a later diagnosis. And we can go into that a little bit later as well, beyond the obvious additional loss of time for intervention, as well as the social isolation and the social ostracism that comes along with being different, especially when you think about girls and boys as they enter middle school and kind of make the transition into high school. So we were, on the one hand, disappointed, on the other hand, intrigued, because of these genetic findings and these findings of girls who were being diagnosed late, both because they were being missed by standard diagnostic channels, and also suggestions that they were able to camouflage their autism. And so that kind of relates back to this notion of doing worse than chance here in panel B, in the sense that, our girls that have a diagnosis of autism, how do they become quite adept at being able to socially adapt, so much so that they're actually have increased activity, somewhat suggesting increased work of the social brain, trying to fit in. And then how does that affect other aspects of brain development for them? Because if you think back to a couple slides ago when I was talking about how the typically developing social brain very effortlessly processes other people's intentions and their actions and helps you fit in socially. In this case, it's suggesting that the girls with autism are accomplishing some of this, but they're doing it at a very high cost in terms of the energy being devoted, quite literally, to their social brain. And what does that pull away from, and how exhausted they must feel, and how does it perhaps relate to a very common observation among studies of girls with autism of diagnostic comorbidity down the road, where greater rates of depression and anxiety and eating disorders in those girls that we see some of in boys with autism, but nowhere near the same type of comorbidity and levels of mental illness that we see in the girls with autism. So on the one hand, a good demonstration of how we can apply advanced data science techniques to begin to identify a biomarker, but then suggesting that much more work needs to be done in order to begin to identify, what is different about girls with autism. And so the University of Virginia is the lead site for a network of locations that are studying girls with autism. They include some really great universities, including University of Washington at Seattle, University of California, Los Angeles, Harvard, Yale, Children's National Hospital, and George Washington University in DC, and of course the University of Virginia. And we're studying girls with autism across this network, in part because one of the strongest kind of biological findings in the field of autism is that autism is much more prevalent in boys versus girls. Now, this is true of almost all neurodevelopmental disorders, in the sense that boys truly are the weaker sex when it comes to neurodevelopmental conditions and neurodevelopment in general. We're much more susceptible to just about every neurodevelopmental condition, psychiatric condition, than girls, but it's even more so in autism, to the extent that when you think about those genetics findings that I mentioned earlier, that girl development, both genetically and in terms of brain development and perhaps social programming, is protective against developing autism. So we needed a network because we needed to recruit as many girls as possible. In statistics or epidemiology, we call that oversampling. And so we're oversampling for girls. We're also studying boys with autism. We're still interested in boys with autism in this network, of course, but we wanted to oversample for girls. And so what we're doing is a mixed methods approach. So the University of Virginia, in addition to being the administrative lead of the network, is also the home of the Data Coordinating Center, and that's actually led by Jack Van Horn. And what Jack is leading is we're recruiting and then taking biological samples, so blood and saliva, and we're taking electrophysiological data, and we're taking functional magnetic resonance imaging data from all of these kids, and then extensive, and actually I should say they're now young adults, older adolescents and young adults. This is a longitudinal study. What Jack's helping us do is bring together all of that data so that we can begin to look at the integration of different levels of analysis of the human organism, from genes to brain to behavior to social context, and understand the bidirectional interactions within and across those levels of analysis over time. And then that, we hope, will lead to greater insights into the development of, on one hand, kids with and without autism, and then sex differences in those patterns of development, and also sex differences within patterns of typical development, okay? So show you one example of where we're sort of going with this. This slide is simply showing you that across our now fairly large data set, when we look at processing of social information, what we see is that in boys, we again replicate this finding that their social brain is different, that they have less activity and less connectivity among social brain regions in comparison to typically developing boys. When we look at girls with autism, those differences are absent, and relative to typically developing girls, what they have is much less activity in regions of the brain involved in processing higher order integration of that information. So it seems, it almost suggests that they're sort of using their social brain, they're overusing their social brain to process this type of information without passing it along to higher order executive prefrontal regions and more action-oriented regions that would allow them to use that lower level social information to begin to process that information and use it in the service of their moment-to-moment interactions. And so that fits very well with, you know, how girls with autism often present, which is that they might be very interested in social interaction. That's in contrast to many boys with autism. And they might be good at certain aspects of it, but then can be terrible at higher level, more subtle aspects of social interaction. So they might overwhelm a social partner and not know when to kind of, you know, sit back and interact less and sort of give that to and fro. And in that can become, you know, alienated and ridiculed for being socially awkward, as opposed to for boys, they might present as kind of uninterested in social interaction. And a lot of times in terms of kind of standards of behavior for boys, that's much less socially damaging to one's reputation than being awkward or bad at it, if you kind of follow my meaning there. So we also began, as I mentioned, to integrate, gene data, so genetic data, in this case, whole exome sequencing. And what we did here was look at the patterns of brain activity in the regions that differentiated girls with autism from typically developing girls during social perception. And we looked at that in contrast to a transcriptome of the human brain that was available from studies of brains totally separate from the subjects we looked at, from prenatal development to early infancy via autopsy, and then what genes are expressed in which parts of the brain. And we made a map that had the overlap between brain regions that we identified as being different in the girls with autism versus the genes that were most expressed in those regions of the brain in early development. And that created essentially a criteria gene set that was limited by location from our fMRI data. And then we looked within that, and lo and behold, what we found was that girls with autism were much more likely to have genetic hits when we looked at their actual whole exome sequencing data in terms of copy number variations, single nucleotide variations. So these subtle genetic changes that were meaningful in terms of affecting a synthesis of a protein. What our data showed was that we could use the imaging data to put a spotlight on which genes that we should be looking at. And then when we turn that spotlight on, we saw this very big difference between the number of genetic hits among girls with autism compared to boys with autism. And ultimately what this suggested was that, what this suggested is that both at the level of brain function and the genetics involved in autism, genetics involved in autisms, the genes that drive girl autism might be very different than the genes that drive boy autism. And the way we got to that discovery was using the imaging data to focus in on which genes we should be paying careful attention to. And we're excited to see that both within our data set and then when we went to a totally different data set, in this case, the Simon Simplex collection, we were able to replicate that finding in the Simon Simplex collection. So we had not only in our ACE network data evidence of this, a discovery of this, but then we had a replication of it in a totally new data set, which of course is a critically important step for anything in science, but in particular when we're thinking about the number of leads within autism that we've seen in our lab and all other labs as well, that then don't pan out in a new data set. It was particularly heartwarming that we could see this in a new data set. So we're excited about that. Now, I focused on social behavior, social perception, but that's not the only story within the field of autism in terms of trying to understand what's going on and what's different in the brains of people with autism. It's really just the lens through which my group has looked most carefully. Another approach to this is an entirely data-driven approach where we don't really apply any preconceived theory, but we say, okay, let's just let the brains tell us what's going on, what's different. One way to do that is through resting state data. And so resting state is exactly what it sounds like. You put the person into the MRI machine, you help them relax, and you tell them either close your eyes and just kind of zone out, or keep your eyes open and look at a little crosshair on the screen. So we're sort of controlling what they're doing and just letting their brains kind of wander. And we measure activity over time and the correlation between different regions of the brain. And we did that in this case. And then we said, okay, what are the underlying latent network organizations that best identify girls with autism from typically developing girls and boys with autism from typically developing boys. And that gave us essentially two maps of the brain and differences in the brain. And then these were statistical maps in these 3 dimensions that said, okay, this part of the brain, this set of coordinates is showing a nice difference between boys with and without autism. And over here, this is a part of the brain that shows a difference for girls with and without autism. We could take that data, that spatial organization, and look in a resource that's online. It's called Neuroscientist, which is it's a really cool website in the sense that you can type in 3-dimensional coordinates of the brain, and it will tell you, it will query all of the available imaging studies that have been mined and for this database, and tell you, what constructs in psychology have been talked about. So if you put in a coordinate that corresponds to Broca's area, for example, in the inferior frontal gyrus, it'll come back with a whole list of studies that have talked about that part of the brain in terms of producing speech, and perhaps mirror neurons as well. And it will rank order the constructs based on how many studies and what level of statistical significance was found within those studies for that region corresponding to that construct. And for us, this was exciting because we could submit our maps and get essentially a diagram of what constructs map onto the different latent networks that we had identified that best defined autism in girls versus autism with boys. And then we turned that into, what's commonly called a Wordle, or Wordles these days. And what you're seeing on screen now, on the top, outlined there in pink, is the Wordle for the girls, their network to construct mapping. And the size of the font, the size of the words, corresponds to the statistical significance of the finding. And then you can see the same thing for boys below it, outlined in blue there. And for boys, this comes as no surprise to anyone who thinks a lot about autism, studies autism. That certainly at the core of the behavioral definition of autism is differences in social perception, in language comprehension, and person perception. And then, of course, those were the most significant constructs that popped out of this analysis. But the data from girls actually really stunned us in the sense that we had to go pretty far down the list before we found anything that corresponded to a similarity with boys, language comprehension being really the only one. And the strongest relations in terms of constructs were anxiety and emotion regulation. And these aren't core characteristics of autism. This isn't, you don't need to have anxiety and emotion regulation difficulties, for example, in order to receive the diagnosis of autism. But for girls, oftentimes it's problems with anxiety and difficulties in emotion regulation that get them to be seen by the proper clinicians who are able to make an autism diagnosis. And as far as parents are concerned, across boys and girls, anxiety and emotion regulation differences are often and difficulties are often the things most cited as being most problematic. This is commonly referred to as autistic meltdowns that are happening well beyond the developmental stage where meltdowns are appropriate. And they cause a lot of trouble for the individual and the family in general, especially as the individual gets older, might be physically stronger and more imposing. These are the kinds of things that get young people in trouble. And so this was quite striking to us and really gave us pause in the sense that what the data was telling us, what the brain activity was really telling us, is that the underlying circuits that are different in girls with autism are different from the differences in boys with autism. And that, kind of at a first pass, even though it's by no means perfect, we could begin to think about autism being reflected in the brain very differently for boys and girls. And that should have obvious implications for how we approach intervention and treatment and this broader concept of precision medicine, precision intervention. So next thing I want to show you is an application of that concept of precision medicine. Having talked a lot about differences between boys and girls, I want to put that away for a minute and turn back to a broader focus of autism in general. And I want to show you an example of where we've taken a well-known evidence-based behavioral intervention. It's called pivotal response treatment. The details of the intervention aren't so important, just knowing that we weren't interested in determining whether or not pivotal response training is an effective intervention. We already knew it was based on the literature, but we wanted to know, could we use imaging to understand how it works? What does it change in terms of the developing brain? And can we begin to predict for whom it will work, okay? And what I should tell you about pivotal response training, like almost every behavioral intervention for autism, it's a very time-consuming and often extremely expensive, especially if insurance isn't paying for it, intervention that requires quite a bit of commitment and time from the family. Typically speaking, in our case, we gave children 16 weeks of pivotal response training, PRT for short, and about 25 to 30 hours a week. So very intensive. Some of that was done in our laboratory at the Yale Child Study Center, and some of it was done in the home. And so this required a parent to bring usually mom, to the Yale Child Study Center, or make their home open to us, meaning that the parent and the child were there. And you start thinking about this, something that could be very expensive. You think about a 16-week course of PRT, it would be costing a family, if they were paying without insurance, about $30,000 for the intervention. With insurance, it would be less, but still a substantial financial commitment. We were giving it for free because it was for research. But if you think about that, that's really, that's quite a burden. And it requires one parent to be available, 20, 30 hours a week, which means that parent really can't hold a full-time job. And of course, for many families, there is only one parent. So you think about all of that, the stakes of being able to predict which kids will benefit from a given intervention become very high. It's not just an academic exercise. If truly we can do a good job at predicting, then we can do something very, very meaningful for families. So this is a complex slide. I'm going to unpack it. The first thing I want you to look at is the different lines on this graph showing pre and post. And the measurement here is the level of social symptoms measured by the social responsiveness scale for each individual child who is in this study. Each child is a black line here. And this is coming into the study, their level of symptoms. And this is after 16 weeks of intervention, their level of symptoms. And what this is showing is a high degree of heterogeneity in how well the kids respond to pivotal response training. So you see that some of them changed very, very little. Going up higher is worse, it's more symptoms. So if they're responding to the treatment well, you see they start here and they go down in terms of level of symptoms. So some kids responded very well. Some really didn't respond at all, and a few got a little bit worse, which is common in all clinical trials. And then the red line is the mean. And when we say evidence-based intervention, usually what we mean is that on average, kids get better, have fewer symptoms, or doing better on our primary outcome measure. And that, of course, was true here. But think about all this heterogeneity. For some kids, this was a waste of time, and for their parents as well, and maybe even an intrusion. And so thinking about this slide, now we look at using these machine learning techniques to identify the parts of the brain that best respond and best predict whether or not an individual will do well. So the parts of the brain that really best help us understand this heterogeneity. These are 4 clusters of activity that help do that. And so cluster 1 is in the posterior superior temporal sulcus. And that's a region mentioned several times. It's involved in really indicating and processing other people's actions and intentions. This is a region of the brain in the parietal cortex that's involved in distributing and maintaining attention. A region of the insula and ventral medial prefrontal cortex, ventral lateral prefrontal cortex, that's involved, cluster 3, widely known to be involved in emotion regulation and being able to regulate your behavior, to engage in an ongoing activity. And this last cluster here, the ventral striatum and the amygdala, very sensitive to reward, including social reward. So finding social interaction rewarding. And what this is saying is that to the extent that these brain regions are active and connected in terms of their activity, the child will do better, that's what this graph is showing, in pivotal response training. Now, a lot of times when I talk about this data, if there are clinicians in the audience, they'll kind of say, yeah, so you just told me that if a child can notice social information and pay attention to it, and if they can regulate their behavior and interact with me, and if they find it rewarding, they'll do well in the intervention that you're talking about. And that seems kind of obvious. It's like, well, yes, but I'm doing 2 things that are deeper than that simple observation. One is these are quantitative measures that vary by child of levels of those constructs that we just mentioned in passing, that really don't have a reliable and sensitive measure out there in nature that can be used by a clinician to make a decision. And in fact, when we looked at all of our behavioral data and all of our clinician ratings, we found that data, in contrast to the brain data, just did a terrible job of predicting which kids would benefit from the treatment. And one of the simplest things we did was ask expert clinicians, do you think this child is a good candidate for this treatment? Can you estimate how well they'll respond? And there's funny things that happen within cognitive psychology talks about these extensively, that when you're an expert in something, it can become very challenging for certain reasons because of cognitive biases to make a judgment about a particular case. And so actually our imaging approach was doing a much better job than even some of the very best clinicians in the country when they reviewed the files and saw videos of these kids, and sometimes met them in person, of being able to predict. So we're doing a very good job of predicting, and that's fine. But we were also showing quite literally parts of the brain that if they were active and connected during the intervention, if we ramped them up prior to the intervention, the hypothesis that we're putting forward is that that would actually allow us to be able to turn a kid who wouldn't have been a good responder into a good responder. And that for us was very, very exciting. So last thing that, next to last thing that I want to show you is another study, separate study that we did where we used oxytocin, which is a molecule that affects social behavior, and it's known to affect the social brain. We gave it to kids with autism. And what we were able to show, this is a double-blind placebo-controlled study, we were able to show that there was a high degree of overlap between the areas of the brain that a single dose of oxytocin would activate and make more interconnected relative to placebo. There was a high degree of overlap between those parts of the brain and the parts of the brain that predict good outcomes and good response to pivotal response training. And so you can sort of see where we're going with this, is that perhaps we've got an approach where imaging can allow us to use a pharmacological agent in a very targeted way for kids who don't have the activity that they need to take part in pivotal response training successfully, to give it to them to boost their response to the pivotal response training, for example. And we think this can apply to any evidence-based intervention, but right now we're actually studying this with funding from the Simons Foundation to see if we can indeed take those kids who don't have the activity initially and help boost it. And I should mention that, we've been talking about kids broadly, and what I'm really talking about is 4, 5, 6-year-olds and above. But of course, autism is a condition that we can see evidence of very, very early in development. So ultimately, we want to push back our ability to study the precursors and the biological markers of autism to the earliest points possible. And so we're using a lot of measures in addition to fMRI, where we're able to scan babies, but we're not able to routinely scan babies. It's not trivial. We can't sedate them. And so we're waiting for them to fall asleep and then get images of their brain in order to allow us to do some of this precision medicine approaches. We have other techniques, including optical imaging and electrophysiology, that allow us to go in and study some of the same concepts with a child who's active and attending, but much, much younger than we're used to studying. So the last thing I want to show you, this is actually being led up by Megan Puglia, who is an expert in working with very young infants. And she's working with the neonatal intensive care unit team here at UVA. And what we're hoping to do is really begin to take some of the techniques that we have and some of the EEG signatures that she has, electrophysiology, and apply it to even very, very young infants. And there, we're not necessarily predicting what interventions would work, but rather, can we predict a diagnosis of autism much sooner and earlier in development so that we can begin to develop interventions for even very young infants so that we can start to affect the developmental trajectory and prevent it from going towards an autistic trajectory even before the onset of obvious symptoms. And so I'll leave you there. Hopefully it generates some questions, and just one last slide to thank the funders of this research and of course the families who have made this possible by allowing us to study their kids and to meet their kids and take these brain images. I'll stop there for questions.
Thank you so much, Kevin. That was fantastic. Autism is such a challenging disorder. And I mean, one of the things which was clearly evident is that what we've been doing up with just, a clinical assessment up until fairly recently has really been inadequate to tell the difference between boys with autism and girls with autism in particular. You mentioned something about the genes for girls possibly being different than the genes for boys with autism. What do we know about the genetics of ASD in the context of it's clearly not one gene, maybe it's multiple genes, if they're sex linked at all, perhaps that's explanatory. What do we know about that? And
Yep.
maybe you can explain some of that.
Sure. So as you mentioned, that there's no one gene that's been discovered that causes autism every time when it's different or present or absent, bigger or smaller. But there are a number of genes that increase the risk for developing autism. And every time I say the number, it's tomorrow it'll grow. But I think the number's around about 100 right now of high confidence autism candidate genes that separately or in combination greatly increase the risk for developing autism. And in addition, so they're kind of high penetrant genes. And then in addition, there are many genes that contribute to risk in sort of the background of common genetic variation that taken in isolation really don't increase the risk of autism. But when you start putting certain common genetic variation together in certain patterns and then possibly combine it with rare genetic variation that is much more likely to directly cause autism, you get this tapestry of many genes. It could be hundreds or thousands that then give you a much increased risk for autism. And even then, it's not deterministic. So you could have a lot of autism risk genes and still not develop autism. You could have one autism risk gene and develop autism. And so it's non-deterministic and very, very complex. And then overlay on that the fact that some of those genes are sex linked, and then that some of those genes play different roles in brain development depending upon whether they're in a boy brain or a girl brain, and the different genetic background of boys and girls. So not necessarily sex linked per se, but in the milieu of what genes do depending on whether they're in the presence of female or male brain development and the hormonal milieu going on, that you get an even more complicated picture. So for example, at first blush from our data, the genes that seem to be hits and candidate genes for boys with autism and autism in general are related to synaptic development, to pruning of synapses, to glial development as well. Whereas in girls with autism, a lot of what we're seeing in terms of the gene modules, gene expression modules that we're finding seem to be more involved with inflammatory response and control and regulation of that inflammatory response.
That's super interesting. So as we kind of reaching the top of the hour here, I'm curious of your thoughts on, are there any data resources that are available for people who would want to, say, take a look at some of these different data types that you mentioned? And finally, just what's the role of data science in helping to make progress in understanding autism spectrum disorders?
Yeah, sure. So, one data resource, of course, is our network. And, since we're the data coordinating center, and Jack, you're the lead of that, being able to collaborate on that data, get access to the de-identified data, and utilize it for different projects is fairly straightforward. In addition, there's more broadly what's called the, well, I call it INDAR. Jack, remind me of what it's now called.
Well, it's the NIH Data Archive.
Thank you. Used to be called the National Database for Autism Research, and it was autism focused. Now it's broader, so they changed the name, but it still contains the autism data from many studies funded by NIH that have contributed to this, are required to contribute to this archive as condition of grant funding. And so you can find incredible, wealth of data in that archive that can be used and integrated, different data types, linked by an individual unique identifier that's still private, but will allow you to know which individual the data came from. And then there also are European similar data sets that are available. EU AIMS is one. I can't speak to how open it is as a resource, but it's there. So those are three at least, and data sets that are readily mineable. And in terms of the role of data science, it's absolutely essential. Any of the methods that I sort of flashed on the screen, EEG, fMRI, behavioral data that we collect, clinical data, genetics, each of those can tell you some cool things in isolation. And of course, data science techniques are used. I highlighted machine learning several times there, but really it's the cross data formats, interactions that data science is so good at generating and handling the giant data sets and bringing together the different types to inform much more advanced biological questions, that where data science shines and is essential to this endeavor. So otherwise it would just be silos of researchers working using their method and publishing the next paper that's kind of logical extension. And that's important and fine, but it's the bridging across all of those different silos and bringing together the data to address advanced questions that is driving the field forward, and going after the most important concepts.
Well, thank you so much, Kevin, for sharing with us your research and your vision for the study of autism spectrum disorder. It is clearly a complex neuroscience challenge. Data science is clearly required to sift through and integrate these multiple different data types. And I hope that some of our participants in our foundation, or our biomedical data science innovation lab activities will look to this as a potential project idea. Anyway, thank you very much, Kevin. Very much appreciate it. Thank you, everybody who tuned in, and we'll look forward to seeing you next week. Thanks so much. Enjoy your weekend.
Okay, thank you.